Assalamualaikum and hi, my name is Nurul Afiqah. In my final year project title is Characterization of Biofilm Forming Abilities in Antibiotic Resistant of Pseudomonas aeruginosa isolates from local hospitals. Glycoproteins are proteins modified with carbohydrate change through glycosylation, a crucial post-translational modification that affects protein conformation, stability, distribution, structure, and function, which playing key roles in physiological processes such as immunity. Antibiotics are pharmaceuticals that combat bacterial infections by either killing bacteria or inhibiting their growth. They have distinct physical, chemical properties, and varying toxicity levels. Some antibiotics exhibit significant chemotherapeutic efficiency against microbial infections in humans and animals. Pseudomonas aeruginosa is a multi-drug resistance pathogen known for its resistance to multiple antibiotic classes. According to One Health Antimicrobial Resistant Organism in 2022, Pseudomonas aeruginosa has shown increased resistance to imipenem and meropenem. Glycoproteins and proteins contribute to antibiotic resistance in bacteria. Pseudomonas is able to create a mechanism like biofilm formation, enhance the resistance, and allowing drug-tolerant cells to persist, which complicating clinical treatments. The objective of this study are to characterize the antibiotic resistance profile from local Pseudomonas aeruginosa isolates, to measure biofilm formation produced by local isolates, and the last one is to identify the differential glycoprotein and protein expression that may be related to antibiotic resistance. Next, methodology. First of all, grow the bacterial culture in the LB broth. After that, strip on the nutrient agar to get a single colony. Using the same nutrient agar, it will be used for gram staining. Next, antimicrobial susceptibility test using this diffusion assay. In this experiment, we use 4 antibiotic discs which are meropenem, aztreonam, penicillin, and ceftazidime. After that, we conduct a 69-well microtiter plate assay to observe biofilm formation of local isolates. Next, we perform protein extraction and separation for sample preparation in 1D SDS speculator. Using protein sample extracted, it goes through protein quantification and purification using Bradford assay and acetone precipitation. After that, protein sample will undergo the separation by 1D SDS batch. Lastly, the gel containing sample will incubate in ProQ Emirate 300 glycoprotein stain, and after that, cyproruby protein stain for imaging and visualization. Next, let's go to the results and discussions. Pseudomonas aeruginosa is confirmed as a gram-negative bacterium because it does not retain the crystal violet stain. Instead, after decolorization, it takes up the contour stain safranin, which give it pink or red appearance under a microscope. Next, variable biofilm forming abilities of Pseudomonas aeruginosa isolates. Two different strains of Staphylococcus epidermidis used as positive and negative control. Out of 10 samples, only sample P2 show a very strong biofilm producer. Sample P6 and P7 were moderate biofilm producer, and sample P1, P3, P4, P5, P8, P9, and P10 were weak biofilm producer. These were determined using calculation in the figure 3 below. As mentioned previously, in this diffusion assay, we use 4 antibiotic Ds which are aztreonam, ceftazidime, penicillin, and meropenem. According to the table 2, all 10 samples of local isolates have low range of inhibition zones starting from 1 mm to 13 mm. This does not meet the standardization regulation set up from Clinical and Laboratory Standard Institute. In a nut case, all 10 samples local isolates were resistant to all 4 antibiotic Bs, which strengthening their title as multi-drug resistance pathogen. Next, protein and glycosylation analysis through 1D SDS patient. In this experiment, we used two stains which is ProQ Emirate 300 and cyproruby. ProQ Emirate 300 is selective for glycoproteins because it binds to the carbohydrate molecules on these proteins, staining only those with attached carbohydrate groups. Therefore, it results in fewer bands on the gel compared to cyproruby, which stains all protein regardless of glycosylation. When used together with cyproruby for total protein detection, the ProQ Emirate 300 stain bands should be as sharp as those stained with cyproruby, indicating well-resolved proteins and precise staining. Candy from the glycoprotein stain kit acts as a protein ladder and appears differently in ProQ and cyprostain, highlighting its use in characterizing glycoprotein molecular weight. Unlike the benchmark protein ladder, each band appears clearer with cyprorubistine than with ProQ. P1 gel A and P10 gel B from ProQ both have lower bands presence, making it hard to detect. Meanwhile, in cypro, both P1 and P10 have a clear line with sharp bands. Overall, beads in cypro are sharper and have better resolution than in ProQ. Possible factors that affect band sharpness are inappropriate gel electrophoresis protocol, full sample preparation handling, and staining protocol. Next, possible reasons of unexpected result of ProQ are due to the technical error, high detection sensitivity, and presence of glycan mimicking structures such as lipopolysaccharide. At the right side, in the area that have been enlarged, we can observe that there are two additional bands on P5. Each of them have molecular weight of 32.3 kilodalton and 31.1 kDa. Based from Uniprot website, possible proteins are HTH type transcriptional regulator and beta-lactams enzyme. Both proteins are related to efflux pump and beta-lactams antibiotic resistant. This would be proved in the distribution assay where sample 5 have the lower less inhibited zone towards for antibiotic dis. In conclusion, all Pseudomonas aeruginosa samples were resistant toward Aztreonam, ceftazidime, penicillin G, and meropenem, thus prove that these samples were multi-drug resistant bacterium. P2 was determined to be a very strong biofilm producer, P6 and P7 were moderate, P1, P3, P4, P5, P8, 9, and 10 were weak biofilm producer. Some protein and glycoprotein changes were identified to be related to antibiotic resistance. Here are some recommendations to prevent any errors from recurring. Use more antibiotic Ds to increase the characterization accuracy of antibiotics resistant profile. Use appropriate and enough antibiotic Ds for all samples isolates. Optimizing sample preparation before performing 1D SDS patch. Optimizing staining and detection protocols, for example, improve washing steps. The last one, repeat experiment and duplicate the data until achieve acceptable and expected results. This is the last one, which is the reference. So thank you for listening.